For this last portion of the chapter, we're going to talk about a couple of things that will impact transcription translation. First one being mutations. We've always heard that mutations, a change in the genetic sequence, the AGCT sequence of the DNA, can cause lots of problems. But we never actually go into detail about those problems. So that's what we're going to do here and now. We're going to look at first at what is referred to as point mutations. Changing one nucleotide. I mean, you sit there and think about it. One nucleotide out of the billions and billions and billions that make up all your chromosomes. How bad could that be? Well, remember sickle cell. Sickle cell was due to the change in one amino acid. One amino acid changed the primary structure, changed the secondary, changed the tertiary, which led to the beta globulin subunit of hemoglobin. Instead of giving you a hemoglobin molecule, 2 A's, 2 alphas, 2 betas, that was free floating in the cell. Well, this altered beta would glom together, forming crystals that would grow and would distort the shape of the red blood cell, giving it that sickle shape. CTC became CAC. You would think, not a big deal. I mean, because, it's A and the T, they're still there. But when you look at the messenger RNA, that codon should be GAG, calling for glutamine. But that change in the messenger RNA from GAG, from GAG to GUG, gives you valine. Doesn't sound like a big change, but it is enough that the primary sequence has changed, the secondary, the tertiary, the quaternary, and you're going to completely messed up red blood cell and all those developmental issues and long-term health issues associated with sickle cell disease. Other mutations, a single nucleotide pair, or complete insertions or deletions. If you have substitutions, you could have a complete pair. Instead of the AT being right there, you have GC. Will that be a problem? Don't know. Is it the first, the second, or the third nucleotide in the codon? You have to see. Sometimes that, substitution, if it's the third position, it's silent. Because remember, wobble. Many of the amino acids have, more than one codon that will call for them. And if you look, the difference in the codons calling for that single amino acid, it's going to be the third position. And those that, the third position change will result in a different amino acid. Well, it turns out those amino acids, evolutionarily, thankfully, those amino acids are still closely related. They're still going to be hydrophobic. They're still going to be hydrophilic, acidic, basic, whatever. So while the primary structure will be changed, ultimately the secondary, the tertiary, and the quaternary structures will still remain. The mutation is silent. A missense mutation will call for a completely different amino acid. Completely different, not related. You still get a full polypeptide, but the shape has changed. Thus, the function of that polypeptide, that protein, will be altered. And the worst ones are the nonsense ones. This calls for one of those stop codons. The mutation actually ends up giving you a stop codon, which will terminate translation early. So you get an abridged polypeptide. You never get a full polypeptide, thus there's no chance to see if there's even going to be any type of function. So when you look at, say here, this example of a silent, you look at, the messenger RNA sequence translated will give you methionine, lysine, phenylalanine, glycine. But that third position, as we see right here, gets changed, which should be CCG, now is CCA. Thanks to wobble, GGC gives you glycine, GGU also gives you glycine. The mutation is silent. We also can have insertion and deletions. Insertion of a nucleotide pair or deletion of a nucleotide pair. These will result in what are referred to as frame shift mutations. Frame shift is an issue. I mean, look at that last bullet there. Insertions or deletions outside the coding part of a gene could affect how the gene is expressed. Frame shift says an insertions or a deletion outside the coding part. Notice how I've clipped off the first letter and added in the first letter of the next word. Insertion or a deletion outside. All the letters are still there. I have now just shifted everything because I have lost the first I. Deletion. Go back to the wild type. The wild type, methionine, lysine, phenylalanine, glycine. You insert an amino acid. You insert a nucleotide pair. So instead of TAC, TTC, AAA, CCG, TAC, TTC, AAA, TCG. Ruh-roh, Shaggy. You have now called for methionine, leucine, phenylalanine, serine. Completely different amino acid. Now, if the next codon wasn't a stop codon already, what you would end up with is a ribosome that would continue on making a new extended polypeptide that from that point, from that phenylalanine on, beginning with that serine, would be gobbledygook of amino acids that may or may not be able to get a secondary structure, but even if they did, it would make no sense and it would have no effect, and this protein would be garbage. How do these mutations occur? There's so many ways. There are numerous mutagens out there, physical, chemical, that you are exposed to on a daily basis. Some of these mutagens can be metabolic byproducts of the food you eat, you drink. Some of them are man-made chemicals that you come into contact with. The worst ones we collectively call carcinogens. Just coming into contact with them will lead to mutations that will lead to cancer.